go through today. We're going to go through the regulatory requirements in the USP, and then we're also going to talk about some of the other systems that are out there for the bacterial endotoxin program and testing. There's quite a few systems that you could use. They all meet the requirements stated in the USP, and we're going to go over all of those systems, and then we're going to talk about some of the nuances of them. Then we're going to talk about some of the other regulatory requirements as part of that as well. So I hope everybody gains a little more information on this. I've worked with several of these systems and have implemented these systems in a couple of different companies. You'll get to understand them and see how they work. You'll find what's best for you in the long run. So let's get started. So the purpose of this webinar is to provide an introduction to bacterial endotoxin testing as described in USP and present do's and don'ts in regards to testing. So there's a lot of little nuances with this test. It can be a little tricky at times if you use some of the older systems that are out there, but for the most part, once you kind of get into an understanding of how they work and how the testing works, you can pretty much boil it down and kind of nail it down and try to dial in. So it's really good. You're going to have some, if you haven't started this testing yet or you are familiar with it, you'll learn a couple of tricks, a couple little do's, a couple little don'ts, and then you'll learn to get it, and you'll get it pretty well in. But it takes some time. This is one of the most complicated tests that I have my analysts do. It's not an easy test to do. It's usually the last one that I have them do after they've proven to me that they can do other tests within my lab. So today's agenda, we're going to go over some regulatory references, particularly USP 85. We're going to talk about what endotoxins are, where they come from, why they are important, how the assay works, and then we're going to talk about some of the methods used for testing. We're going to talk about choosing the best assay for your product, some do's and don'ts during assay execution, and then we're going to have a review, and then we'll have a Q&A section. Of course, all of this is this is our guide. We may talk about some other things as well, but for the most part, this will be the topics that we're going to discuss. Okay, so the regulations. The first regulation we're going to talk about USP 85. That's the governing regulation for bacterial endotoxin testing, and it also tells you why it's important. Some of the things that can occur if you do not test for bacterial endotoxin, especially now that we're using, a lot of companies are using E. coli cells for their manufacturing of their drug product. So it's very important to ensure that you have that bacterial endotoxin at an acceptable level and within your product. And how are you showing that you are removing that throughout the process? Remember, E. coli cells have this endotoxin within their cell bodies or their cell walls, so you need to make sure that you're able to remove it throughout your process, throughout your downstream process. We'll also talk about the European Pharmacopoeia chapter 2.6.14. There aren't too many differences between these two chapters. They pretty much have adopted the same methodology when it comes to bacterial endotoxin testing. They're pretty much the same. There's not much difference in terms of chapters. Usually the European Pharmacopoeia is a little different in some testings, but when it comes to bacterial endotoxins, the USP is pretty much what it follows. Okay, so here we talk about endotoxins. Endotoxins are large molecules consisting of a lipid and a polysaccharide composed of an O antigen. So here we have the O antigen here. Here we have the polysaccharide, and then it's joined by a covalent bond that are found in the outer membrane of gram-negative bacteria. So one of your most common ones that you'll see, especially if you're using E. coli for production of E. coli, of course, but some of the other stuff as well. Pseudomonas is another gram-negative bacteria that can contain endotoxins. So you'll see that it may develop within your water systems. It's a hydrophobic a hydrophilic organism that loves water. It likes to sit in the water systems, create a biofilm, and you may see it in there. So that's why a lot of times they have requirements for testing your whiskey, your clean steam, is to test for endotoxin. So, and the purpose of this whole endotoxin chain here is that it elicits a strong immune response in animals. Okay. And what I mean by a strong immune response is it usually produces a fever, flu-like symptoms, stuff like that. Okay. What